This week we start our roundup with virus-related news. Sanofi Pasteur, the vaccine's global business unit of Sanofi, externalized COVID-19 vaccine research and inking an extension deal with Translate Big, a clinical stage messenger RNA or mRNA therapeutics company to develop a novel mRNA vaccine for COVID-19. Now, this class of technology compares with other ventures developing mRNA-based approaches, including Moderna. The two already partners leveraged existing agreement memorialized in 2018 in place for the development of mRNA vaccines for infectious diseases. Translate Bio has developed multiple NRA constructs on its mRNA platform to discover, design, and manufacture SARS-CoV-2 vaccine candidates. The large French company offers the deep expertise and support from external research networks in a bid to accelerate the advancement of vaccine candidates. Now, already with a deal underway with the Biomedical Advanced Research and Development Authority, or BARDA, to advance a COVID-19 candidate, recombinant protein-based vaccine candidate targeting COVID-19. Now, this second focuses on a prospective approach using a different technology and, of course, vendors. Now, both companies are targeting mRNA-based vaccine development for COVID-19 that transport genetic information from DNA to ribosome, where it dictates an amino acid series that produces proteins. The hypothesis goes that by intelligently engineering a patient's own mRNA, there is a potential to use it to engineer cells and transform them into protein production centers, producing specified drug molecules associated with new therapies. Now, although Moderna is the most well-known player among this biotech class, it isn't alone. It's joined by a BioNTech partnered with Pfizer, Translate Bio aligned with Sanofi CureVac, which President Trump reportedly approached in a lucrative exclusive buyout deal, which they did reject. Now, these vaccines work by mimicking disease agents to stimulate the immune system, building up a defense mechanism that remains active in the body to fight future infections. mRNA vaccines offer an innovative approach by developing a nucleotide sequencing encoded with antigen or antigen selected for the high potential to induce a protective immune response. We'll keep you posted on this story as it continues to develop. Trial Site News is committed to the movement of clinical research transparency and accessibility. Now, during the COVID-19 pandemic, we expand some of our coverage based on requests from our readers. Now, in the interest of keeping our readers attuned to all levels of the COVID-19 medical crisis, we are reviewing how different municipal entities are handling the issue of testing for virus and/or virus antibodies. In a prior piece, we covered COVID-19 testing in San Miguel County, Colorado. Now, in that small county of 8,200 residents, antibody testing, a more economical and in some ways more useful measure than testing for live virus, is now being utilized to make testing available for all. This was a special case in that the Atlantic magazine reports that, and I quote, "The tests are being donated by United Biomedical, a multinational biotech company whose executives, a married couple, happen to live part of the year-end Telluride, the resort town that is also San Miguel's county seat. A lesson of San Miguel is that pervasive, all-encompassing testing is needed if silent carriers are to be identified and quarantined. Another is that extraordinary resources may be called for." Updated information from the Mercury News on March 28th adds to what we know. Public Health Department released detailed data of COVID-19 cases within the county Friday that provides breakdowns of the ages and genders of people who have tested positive and people who have died from the novel coronavirus. There were 32 new positives on Friday, making the county total 54, second in California only to Los Angeles. The current county death toll is 20. The health department has said that because of limited testing capacity through the public health laboratory, the number of cases that we detect through testing represents only a small portion of the total numbers of likely cases in the county. The County of Santa Clarita Public Health Department said the data from commercial and academic laboratories who report results to the California Reportable Disease Information Exchanges are reflected in this data. The key facts about Santa Clara found so far are that half of the positives have been people under 50 years old. One person under 50 has died. 70% of the deaths have also been men. MedPharmics, a New Orleans area clinical investigational site, recently trained local medics on how to properly administer COVID-19 tests. Medical Director Robert Jeanfrau, MD, believes that the thousands of smaller research sites across the country could form an army of high-impact COVID-19 support resources, directly training medics and other emergency personnel while establishing standard operating procedures and other guidances for how to properly train and guide testing, not to mention support what we can now be considered top priority scientific research. Now, after all, there is no vaccine yet for SARS-CoV-2, the virus behind the COVID-19 panic. Dr. Jeanfrau and his wife, Andrea Jeanfrau, also collaborate with the local hospital, East Jefferson General Hospital, to collect blood samples to support a clinical trial addressing more extreme COVID-19 cases involving severe high-risk cases. Dr. Jeanfrau notes that there is something about an individual's immune system that is somehow possibly connected to why some people get better and others fare worse. In New Orleans, the Jeanfraus are on a mission to employ scientific research to make a difference against COVID-19. Now, during this pandemic, Trial Site News is calling out to our readers' attention the importance and criticality of clinical investigators around the world, whether employed in major academic medical centers, specialized nonprofit institutions, industry sponsors, or private practices, such as Dr. Jeanfrau. Now, who are the heroes in this age of pandemic? Well, the research scientists are working tirelessly around the clock in search of a vaccine or a cure. Research is more than just incredibly important at this point. It would appear that the virus is gaining steam into Louisiana and New Orleans. Now, according to recent reports, Louisiana now has 2,746 cases and 119 deaths, while New Orleans reports over 1,000 cases. IQVIA and Quest Diagnostics joint venture, known as Q2 Solutions, which focuses on clinical trial laboratory services, has announced a collaboration with the University of Texas Medical Branch, or UTMB, to develop a novel assay for COVID-19 tests, a key tool necessary for an ultimate vaccine. Now, Q2 Solutions will be mobilized for the research into COVID-19 vaccines. UTMB has developed a novel reporter COVID-19-based test which could offer some advantages such as greater sensitivity and considerable improvement in assay throughout as the assay time moves to a single day from many days. Now, UTMB professor of human genetics Paiyang Shi reports that they have successfully produced a reported virus system engineered with either luciferase or fluorescent tags to enable quantitative determination of vaccine effectiveness. Now, he said that this test will enable Q2 Solutions to test sera from individuals participating in vaccine clinical trials to see whether the vaccine has induced antibodies that block infection of the virus, and thereby answer critical questions in the vaccine development process. Leading clinical trial observatory services with bioanalytical, genomics, vaccines, flow, symmetry, anatomic pathology, immunoassay, companion diagnostics, and central laboratory services with secure enterprise-wide biospecimen and consent management solutions. University of Pittsburgh School of Medicine researchers scientists have a potential vaccine targeting SARS-CoV-2. Preclinical research on mice revealed that the fingerprint-sized patch produces antibodies specific to the novel coronavirus at quantities thought to be sufficient for neutralizing the virus. Now, recently, when tested in mice, the experimental product known as PIT-CoV-AC generated a surge of antibodies against SARS-CoV-2 within 2 weeks of the microneedle pick. The University of Pittsburgh team was previously involved in vaccine research for SARS-CoV in 2003 and MERS-CoV 2014, both closely related to SARS-CoV-2. And it involves the spike protein, which researchers have learned is important when considering inducement of immunity against the virus. The University of Pittsburgh-based researchers subjected the underlying product to significant external peer review. They highlight to the experimental mRNA vaccine candidate now in clinical trials, mRNA-1273 from Moderna, noting that PIT-CoV-AC, short for Pittsburgh Coronavirus Vaccine, follows a more established approach. For example, using lab-made pieces of viral protein to construct immunity. The team designed the vaccine to build on the original scratch method used to deliver the smallpox vaccine to the skin, but in a more high-tech way that is more efficient and reproducible patient to patient. The researchers also used a novel approach to deliver the drug called a microneedle array to increase potency. Now, this array is a fingertip-sized patch of 400 tiny needles that delivers the spike protein pieces into the skin where the immune reaction is strongest. The patch goes on like a band-aid, and then the needles, which are made entirely of sugar, and the protein pieces, which simply dissolve into the skin. Now, as reported, the experimental vaccine has worked on mice. However, the animals haven't been monitored long-term as of yet, but the investigators can show presently that those mice who received the MERS-CoV vaccine produced a sufficient level of antibodies to neutralize the virus for at least a year, and thus, for the antibody levels of the SARS-CoV-2 vaccinated animals are following a similar positive trend. The researchers noted in their press release that the SARS-CoV-2 microneedle vaccine maintains its potency despite sterilization with gamma radiation, a key step towards producing a suitable product for human intervention. And now we turn to autoimmune-related news. Pandion Therapeutics announced the closing of an $80 million Series B financing to support the continued development in Pandion's pipeline, including lead clinical stage program PT101. The financing round was led by Axis Biotechnology and Boxer Capital and included other new investors, RA Capital and OrbiMed. Now, in addition, all of Pandion's existing venture capital investors, including Polaris Partners, Versant Ventures, Roche Venture Fund, SR1, JDRF T1D Fund, and Bioinnovation Capital, participated in the round. Pandion's pipeline includes leading clinical stage program PT101, a novel interleukin-2 mutein Fc fusion protein, which is currently in phase 1 clinical development to assess the safety, tolerability, and pharmacokinetics of PT101 after subcutaneous administration. The clinical trial will also include regulatory T-cell expansion and other pharmacodynamic measures. And the financing is also expected to aid in bringing a second program into the clinic and broaden Pandion's modular drug design. Do you have any clinical news you'd like us to report on? If so, let us know. Contact information is in the box below. Thank you for joining us for this week's Trial Site News Roundup, and remember, subscribe and ring the bell to be notified of updates, and we'll see you next time.